Hello, this is Kavita Kundra, and today we are presenting Mscdfsm Biochemistry, Lipid and Protein, Part 3. Nucleic acids. What are nucleic acids? Nucleic acids are the high molecular weight polymers present in all living organisms. There are two types of nucleic acid we all know, DNA and RNA. DNA means deoxynucleic acid and RNA is ribonucleic acid. The monomeric unit are called nucleotides. Complete hydrolysis of nucleic acid yield heterocyclic nitrogenous base, 5 carbon sugar and phosphoric acid molecule. So if you complete hydrolysis means if nucleic acid fully break down, then what it will generate? It will give you a nitrogen base, carbon sugar and phosphoric acid. Function. What is the function of nucleic acid? Function is storage and transmission of genetic information from one generation to another generation. Amino acid structure, function and structure, classification and properties. Amino acids are the building block of protein. Amino acids are the compound containing NH2 amino group and carboxylic group COOH. Normally 20 different amino acids are found in protein. What is the journal formula? It is very important. R bonded by CH and it is further bonded by COOH, that means carboxylic group, and down it is bound by NH2, that is amino group. R represent the side chain that varies according to the amino acid. And types of amino acids with non-polar side chain. It is all because of side chain. There are different types of amino acid. One is non-polar side chain, one is polar but uncharged side chain. Uncharged means there is no charge, no positive, no negative. Amino acid with positively charged side chain and amino acid with negatively charged side chain. Amino acid with non-polar or hydrophobic side chain. What are they? They are 8 in numbers. This is the formula I have generated. ALIM PAP TV. So this is the name you have to remember. Alanine, L for leucine, isoleucine, M for methionine, P for phenylalanine, A for PA is phenylalanine. It's the same. PA is same. And proline, T for tryptophan and V for valine. So you have to remember like this. So it is easy to remember. LM PAP TV. Amino acid with uncharged polar hydrophilic side chains. They are less soluble in water than polar amino acid. Hydrophobicity increases with increase in side chain. Amino acid with uncharged polar side chain. These are the neutral polar functional group and able to make hydrogen bond with water. The polarity of serine and threonine is due to OH group. The polarity of asparagine and glutamine is due to CONH2 group. And examples are STT, GG, AC. S for serine, T for tyrosine, T for threonine, G for glutamine, G for glycine, A for asparagine and C for cysteine. Amino acid with positively charged side chain. Lysine, histidine, arginine. So you can make the formula as HAL. Amino group of lysine and guanidine group of arginine are responsible for positive charge. You can ask in examination you can get the question like 1 mark question, how these positively charged amino acid get their positive charge. So you can say amino group of lysine and guanidine group of arginine are responsible for positive charge. Amino acid with negatively charged side chain, there is only examples, no need to go deep. Aspartic acid and glutamic acid. Properties and chemical reactions of amino acid. In properties, first we'll discuss physical property, solubility, melting point, taste, acido-basic property, stereochemistry of amino acid, hydrophobicity and absorption spectra. So there are 7 different physical properties. Solubility. Amino acids are readily soluble in water, slightly insoluble in ethanol and insoluble in ether. Tyrosine is soluble in hot water. Cysteine is soluble in hot water with difficulty. And hydroxylproline are soluble in alcohol and ether. Amino acids are generally soluble in acids and bases. And then it's a melting point. Amino acid have high melting point, means you have to uh, give them the heat of 200 to 300 degrees, then only amino acid will melt. Taste, they are sweet also, they are tasteless also, they are bitter also. In sweet, what are the sweet amino acid? Glycine and alanine, they are the sweet one. And leucine is tasteless and arginine is bitter. Monosodium glutamate. What is MSG? MSG is very important topic. It's very important ingredient. Sodium salt of glutamic acid is a flavoring agent for certain foods such as sausages. It imparts flavor. Since 1908, it is discovered by Japanese chemist Kikuné Ikeda. The studies have also shown that it causes cancer and brain damage. For a healthy adult of 60 kgs, only 7.2 gram is accepted. So you have to be very careful whenever you are using, if you're having food systems and if you're having some uh, cooking centers or anything, you have to be very careful. You just have to use very small amount of MSG. Acido-basic property. Depending upon the pH of the solution, amino acid ionize and act as weak Bronsted acid or base. Means either it will be acting as proton donor or proton acceptor. This ionization facilitates their quantitative analysis. All amino acids may be an acid or base. Amino acid, because they contain both acidic and basic group. Acidic group is COOH, basic group is NH2. Because of this unique structure, amino acid have characteristic of acid and base and are reacting chemically either as acid or base, and they are said to be amphoteric. So you can have question like why amino acid is amphoteric, because they contain both acidic and basic group. When both the amino and carboxylic group get ionized, it is said to be zwitterion and behave as neutral compound. Any compound that has net zero charge is called zwitterion, and this is an amphoteric molecule which behave as acid and base, and the pH is known as isoelectric point. So what is zwitterion? Zwitterion is a compound, any compound that has net zero charge. An amino acid is also a zwitterion because it has no charge. It can act as acid or base because it has carboxylic group and amino group. What is the stereochemistry of amino acid? All amino acid except glycine rotate the plane of polarized light because of the presence of asymmetric center at carbon number 2. When amino acid is at left of C2, it is called L form. All amino acid in diet exist in L form, LEVO form. Hydroxyproline and hydroxylysine have second asymmetric carbon. Thus, four stereoisomers are possible. When optically active amino acid react with alkali, it get converted into racemic mixture. Then is hydrophobicity. Hydrophobicity of amino acid determined by the relative solubility of amino acid in water and in less polar solvent like ethanol. They are grouped as hydrophilic and hydrophobic depending upon the side chain, whether side chain is hydrophilic or hydrophobic. So it's all depend upon the side chain. If the side chain is hydrophobic, the amino acid is said to be hydrophobic. If the side chain is hydrophilic, they are grouped as hydrophilic. So example of hydrophobic is tyrosine, and the example of hydrophilic is arginine. NaSN. Absorption spectra. All amino acid absorbed at the wavelength of 210 nanometer. Tyrosine and TYR are the only amino acid that shows natural fluorescence. They have natural fluorescence, natural light. Chemical reaction of amino acid. In chemical reaction, there are three types. Like when the carboxylic group react with something, that is also chemical reaction. Then amino group will react, that is also a chemical reaction. And then third part is side chain. So first we'll see reaction of carboxylic acid. Salt formation. COO group, COOH means carboxylic group react with metal hydroxide to form amino acid salt. Ester formation. Amino acid react with alcohol in presence of dry HCl and it forms ester. This ester as well as free amino acid can be reduced to the corresponding alcohol with lithium aluminium hydride. Next is decarboxylation. Amino acid undergo decarboxylation reaction by treatment with alkali, heat, acid to form amines. These amines have biological activities like histamine mediates allergic reactions. Histamine and tyramine has pharmacological properties. Reaction of amino acid. Reaction in under reaction of amino acid, one is ninhydrin reaction, another one is Sanger's reaction, and another one is Edman reaction. Ninhydrin. What is ninhydrin reaction? It is the powerful oxidizing agent. When ninhydrin react with amino acid, oxidative decarboxylation result in the formation of carbon dioxide, NH3 and aldehyde. The reduced ninhydrin reacts to NH3 forming a blue-purple complex which has maximum absorption at 570 nanometer. What is Sanger's reaction? This reaction is specific for amino group of amino acid or peptides 1-fluoro 2,4-dinitrobenzene FDNB. You can get the question of asking full form of FDNB. So it's 1-fluoro 2,4-dinitrobenzene. Results in the formation of DNP derivative, which is yellow crystalline compound. Edmonds reaction. When we need to identify amino acid at N terminal of peptide, this reaction is used and phenyl isothiocyanate. Phenyl isothiocyanate react with amino acid to yield cyclic product called phenyl thiohydrantoin. Reaction of side chain. Reaction of thiol group with iodoacetic acid. Reaction of either group of group with formic acid used to determine methionine as methionine sulfate, reaction with p-chloromercuric benzoate to determine cysteine, and reaction with Alman reagent to determine cysteine. These are the few reactions, and this topic is over with these reactions, and you can go through it thoroughly. And if you have any questions regarding anything, you can ask me. And thank you so much for your time. I hope it will help you in your studies, and I wish all the best for you all. Thank you so much. Stay safe, stay protected.